So quick recap of all that we have learned till now. Will you please do that, Aishashi? So that the new students will also know about what all we have studied till now. Alina, will you?
Yeah, ma'am. We studied about reproduction
Mm-hmm.
and making of curd, formation of curd, and about mitochondria.
Mitochondria, no. We read about there are two types of reproduction. One is sexual and another is asexual. And sexual reproduction, we have seen that we need two gametes, male and female, for sexual reproduction. Whereas for asexual, there is only one. And in asexual reproduction, we have seen through budding. Budding is one method through which we have seen how an organism reproduces through a budding. The whole life cycle of budding, you have seen how, a bud is formed, and then from the centriole, how it gets divided into two daughter cells. And then we have seen about fragmentation, one more method of asexual reproduction, that is fragmentation. And in which animal have we seen that fragmentation? Yes. Which organism is this?
I think this is mitochondria.
This is not mitochondria, is the powerhouse. It's an organelle inside the cell. This is a different organism. Mitochondria is an organelle, which is also called as the powerhouse of the cell, which is responsible for the process of respiration in an organism. But this is a complete organism. Inside it, there are many organelles like mitochondria. So what is the name of this organism? And this organism, yesterday we have discussed that through asexual mode of reproduction, the reproduction takes place in fragmentations, where a part of it, as I told you, a part, or like your fingers are also divided into 3 fragments. So you can just see that one part of a finger falls down and it grows into a new organism. So this is Spirogyra. We were talking about this organism. It's called as Spirogyra. So you can see what happens in Spirogyra is a flat worm. It's a flat sort of worm, and even you see this molds, leeches, Spirogyra, they all grow in this manner. It's not only that Spirogyra where it happens, you can even see in the flat worms. Okay, and molds are also there. Molds mostly we see the fungus बूझ आ गया बोलते हैं ना हम लोग जो कोई चीज के ऊपर if you are keeping it for a long time outside. So that is nothing but molds. And even in the lichens you generally see on the trees. It is also a sort of organisms, that eats away the bark of a tree. And we even see in Spirogyra. So all these organisms grow through a process called, what was that process? What is this process called? Yes. This is the process we are talking about. What is this process?
Fragmentation.
Fragmentation. So through a process called fragmentation. So all these organisms multiply, reproduces through fragmentation process. Now, if you can see, they also reproduce sexually. So fragmentation is a common mode of reproduction in large fungi and land plants, like, even you can see it in mushrooms. And there are a way through which we can even do artificial propagation. What do you mean by artificial propagation? Can anybody tell me? What do you understand by the process called artificial propagation? Yes. What is artificial propagation?
Cutting, layering.
What is this cutting, layering? किस पे कर रहे हैं हम लोग? On which organisms are we trying it?
Plant.
Plants, yes. So what we are doing is through an artificial method also, this reproduction is, you know, that regeneration that takes place. So regeneration takes place of the duplication of the organisms takes place. So what we are doing is, that is asexual or sexual. We have seen that budding, fragmentation, these were the methods where, you know, it were natural methods. They were not artificial. They are happening themselves. Natural is organic by itself. Nature, naturally it is happening. Like in something we can do it artificially also. Human beings are also helping, you know, we human being think, because, you know, as you all know that as the population is increasing, there is a need or high demand of food also for the people. So what we did is, naturally produced food is not sufficient. So the science has developed and thought of making sure that every organism that lives on this earth gets its food. For that, the scientists had thought of coming up with the artificial methods, artificial way of propagation, and you know, getting food, for making food more common and available for everybody. So what is we doing is, the first artificial method is cutting. In this cutting, what are we doing is, we call it as vegetative propagation in plants, in which, you know, new plants are developed from the cutted portion of the existing plant. So write down this definition of cutting. And what do you mean by artificial method, vegetative propagation? And after you write this sentence, I want each one of you to read it out and explain me what do you understand from this statement. Of the method of artificial propagation in the plants is cutting, and there are many more methods. Let's see the next method also, by which how can we artificially propagate plants. And you can just see that how a cutted part of a plant has developed into a new one. And some plants grow individually when a piece of parent plant having bud is cut from the existing plants. So when you have to cut, you have to make sure that these are called buds. You can see these red colored buds. These buds develops into the flowers. So if you are cutting it, if you are cutting a part of a plant with the buds, then only you can see that it develops into the same like a you know, parent plant. A new plant that grows becomes into a parent plant. If you are just cutting a part where buds are not available, then you may not be able to see that. And after a few days, if you cut the parts having buds, and this part again grows into a new individual plant. Like this, generally we can see in the plants like rose and hibiscus. And then the second method of artificial propagation is called as a layering. So layering, Aisha Sheikh, can you please read it out and tell me what do you mean by layering?
A branch of the plant
Branch of the plant, yeah.
with at least one node is pulled towards the ground and a part of it is covered with moist soil, leaving the tip of the branch exposed above the ground.
Yes, so you can see that what's happening in this layering is, if you can see this spotted part, you know, this one, this is pushed down on the ground, okay? And then this develops into, once it is going down the ground, we can see that it develops roots here after going on the ground, as soon as we have learned about geotropism and phototrophism. So geotropically, roots develop towards inside the ground, and this again develops into a new plant. So let us see how layering, artificial propagation is possible through layering. So this is again artificially done. We are forcibly putting it down to the towards the ground and allowing it to grow into a new plant. So you can see how the layering is happening. And then, you know, in the once it falls down on the soil, it develops roots over there, and this develops into a new plant. After some days, you see how the roots are developing into to that part of the branch which is buried inside the soil, and this branch starts growing, develop and grow and becomes into a new plant. That's how a new plant is formed. So this is also another way of artificial propagation, and this happens, example is nerium. And then the third interesting method is called as grafting. In grafting, there are two terminologies which is very important, which you have to remember, and it is called scion and stalk. So during grafting, what are we doing is two plants are joined together in such a way that these two stems joins and grow as a single plant. We can see that a part of this plant and a part of this plant, they both are cut, and they are from, you know, this is, for example, this is the parent plant, and this is a new daughter plant. So apart from a parent plant is cut and it is attached to a daughter plant, and so that we can get a desired offspring. So this method is called as the grafting method. So this grafted one produces into a new plant which has got both the varieties which are mixed together. So the scion, what is scion and what is stalk is important, which is generally asked in your exams, and you should be knowing how it happens. So in grafting method, what are we doing is stalk is already existing to the plant to which the scion is added. Scion, we are getting it from other plants. You know, the new plant on which a part of parent plant is added is scion. And stalk is already the plant on which we are attaching this scion. So please note it down. And then I want Aisha to read and explain this. Aisha Sadaf Khan. Yeah, Aisha, will you please read it and explain us what are we doing here?
One which is attached to soil is called stalk, and the cut stem of another plant without roots is called scion.
So we have seen that how, you know, the one which is attached to the soil, this is the stalk, and to which we
Yes.
are attaching. So this is called scion, and that is how this develops into a new plant. So this method is called as grafting.
Okay, ma'am.
Yeah. So everybody understood. So in grafting, both stalk and scion are tied with the help of a twin thread and covered by a polythene cover. So that is how, you know, we artificially they are tied fixed, and then with the polythene cover it is covered for some time, and later that grows into a new plant. And grafting is used to obtain a plant with desirable characteristics. So what are the desired characteristics that we can see? So plant in which grafting is done more routinely are mango, apple, citrus, etc. So most of the we do see so many varieties of mangoes, so many varieties of citrus fruits, oranges, and you know, many more citrus. All khatta fruits are called as the citrus fruits. And apple also we see so many varieties. Why do we see? Because we are using different scion and the stalk, and through this grafting propagation we are cultivating many varieties of fruits and vegetables through this process. Now, another method of artificial propagation is also through spore formation. What happens in this spore formation is this is also an asexual method of reproduction, which occurs through microscopic reproductive units called spores. So mostly in rhizopus or mucor, bacteria and non-flowering plants such as ferns and mosses, they reproduce by a method of spore formation. So how is this reproduction takes place? Let us see. Through spore formation. You can see that most of the fungus you see, that is pura booch, jisko hum booch bolte hain. If you are keeping bread for a long time, you see the molds coming on it. So these fungi and rhizopus, or these are the microscopic organisms, and they are non-flowering plants. They they reproduce by a method called spore formation. We can see that these spores contains so many buds inside it, and this is a life cycle of the fern and rhizopus. And in it, what is happening is sporangium, and you see it as a sporangiophore. So you can see mycelium. These root-like, hair-like structures are called as mycelium, and this whole bud. Are you all able to understand me?
Yes, ma'am.
Yes, you all are with me, no? Right. So this is sporangium, you can see, and this stalk is called as a sporangiophore. And in this, you can see that there is a stolon, and we see type 1 and type 2 mating. These which are produced nearby in this rhizopus, we see that the buds that are growing, the spores that are growing near each other, they meet with each other and they produce into a new daughter or a new offspring. So how is it happening? Let us see. So this burst out, generally what happens is this is sporangium, which you are seeing here. This sporangium bursts out because of the pressure of the air. When there is a wind blows or a pressure of air that blows on them, it bursts into the air. And then what happens is it spreads all the spores outside. It is filled with, this sporangium is filled with spores. You can see these small spores. They are just like a pollen of a plant, which we have seen. So these spores, they are very light in weight and they spread very quickly under any favorable conditions, and they germinate and produce into a new plant. So you can see that the sporangium contains so many. You must have seen it on many of the leaves. At the back of the leaves, it is one near our school also. There is one plant, in that Dr. Dentist Radhika ma'am's house. You can see this plant where, at the back of the leaves, you can see so many sporangiums, and each of these sporangia filled with spores bears, sporophylls, and these clusters that are on the lower side of the leaf, and they burst out and they produce into the new plants. So this is one of the more artificial method of propagation through spore formation. And non-flowering plants like fern and sporophylls, they produce under favorable conditions. Particular climatic conditions are very much required for their growth, and they grows into a new plant. So let us do a lab activity to see how it works. To grow rhizopus or a common mold, you can just observe them into your home also, where you required only a piece of bread, a plastic bag, a plain glass slide, a cover slip to observe under the microscope, a water eyedropper, and a disposable gloves. Gloves are very important because these are infectious things, so you should never touch them with your naked hands. So in this procedure, how are we growing molds? Please listen to me carefully. Aziz is going to explain this procedure after me. So what are we doing is we are taking a soft bread and we are leaving it for some time outside. And then we are sprinkling some water on it and keeping it into the plastic bag. And so that air is not allowed to enter inside. And then we check this piece of bread after a few days, it gets dried. Then what are we doing is, again, we are pouring some water and then keeping it back into a plastic mold. Then you find mosses of a black, gray, or green dotted structures that are produced on this bread mold, and we call them as bread molds. Have anyone of you observed this happening in your home? Did anyone saw this?
Yes, ma'am.
Yeah, so how quickly it is. You must have seen that how fast this, if you observe this bread, you keep it for some time, within few minutes and the hours you can see its colonies developing if you can observe it under the microscope. So you can see, if you can remove this, put it into the slide, put some water on it and cover it with a cover slip, and then you observe it under the microscope. So you can see these colonies of molds that are formed. You have to make sure that when you're observing under the microscope, there won't be any air bubble, so that you can see. So you will see this sporangium that is formed. So this is the procedure of making the slide. What are you doing is, you are using a dropper, and with that and with the help of a toothpick, you can take some mold from the bread and put it on this droplet of water. And after that, what are you doing is, you are covering it with a cover slip. This cover slip is a thin plastic slip, which you can see it in the lab. How many of you have seen this cover slips and slides in the lab? Have you ever seen it?
Yes, ma'am.
You have seen them, right? And when we are putting this cover slip, you must have seen that your teacher is cleaning it with a tissue from outside and press it properly so that there is no bubble inside. Because if there are any bubbles that are left inside, you will not be able to see it clearly. Now, under the compound microscope, you can see these molds developing under it. So what are your observations? The common bread mold plant consists of fine thread-like projections called hyphae and thin stem-like knob called the structures or sporangia. And this sporangia again bursts out and spores are and gives rise to hundreds of minute spores that grows into another new sporangia. So this is how it takes place. Now I would like Aziz to explain this procedure. What have we done into this spores formation? How is propagation takes place, reproduction takes place through spore. So waiting for you, Aziz, to explain this. Yeah, Aziz, unmute yourself and explain.
Can you explain once again?
Anybody here in the group who can explain it to Aziz, who feels that they have understood what have we done in this lab activity?
We have to observe the bread cells.
Yeah. Alina, will you please do it in Hindi? You can explain in Hindi so that everybody understands.
Okay, ma'am. ब्रेड सेल्स ऑब्जर्व करना है माइक्रोस्कोप के अंदर. पहले ब्रेड को कोई टूथपिक से थोड़ा ब्रेड का क्रम्स लेके स्लाइड पे रख के we have
हम पहले उस पर हम लोग पहले क्या कर रहे हैं, वो ब्रेड के ऊपर मोल्ड्स बना रहे हैं. मोल्ड्स कैसे बन रहा है? How did we got molds on the bread?
By adding some water.
Very good. We added some water on the bread, and then what did we do?
We added it on a slide.
No, this bread we have kept inside a polythene bag for a day, and then next day again we removed it and we saw that it got dried up, and then again we sprinkled some water and kept it back into the pocket, plastic polythene bag. Then we have observed that there were black and gray, green molds that were developed. Now we are going to observe these molds. So tell us the procedure, Alina. How are we going to observe these molds?
Yes, ma'am. We are gonna observe it in microscope.
Yes. How are we preparing a slide?
Yeah, we are preparing a slide by keeping some of that mold on the slide and covering.
How much mold we are taking? Because one sporangium has got hundreds and thousands of spores. So what are we using to take that mold and put them on the glass slide? What are we
The
using?
tip of with.
Yes, very good. We are taking just a toothpick, a small stick of toothpick. With the help of that, we have taken it. And then what did we do?
We covered it with covering slide.
Yes, we have also put a drop of water on it, right?
Yes, yes.
And then
And then we are gonna observe it.
under compounds microscope. So then you have
Yes.
seen so many rhizopus that has grown there. So this was the lab activity which you have done it. So go through this thoroughly and come back tomorrow for the class. So Aziz, you have to write this procedure. Have you understood now? Aziz, समझ में आ गया?
Yes, ma'am.
So please write it in your own words and post it in the WhatsApp group. We have got just 1 minute left. I don't know, anytime it can shut down. So I want you all to come prepared with all these videos which you are sharing in your group tomorrow. And if you've got any doubts, please feel free to post them or in the WhatsApp group and get them clarified.
Okay,
Okay,
ma'am.
ma'am.
Okay. So take care. See you all tomorrow. Allah Hafiz.